Hi there students, this is Professor Araneta and I will be lecturing on the role of mitosis and meiosis in plant sexual reproduction. So without much further ado, this will be the objectives of my discussions. At the end of this lesson, the student must be able to define essential terminologies relevant to the understanding of plant sexual reproduction and describe the mechanism of alternation of generations in plants, giving more emphasis on gametogenesis and eventual fertilization. Before we proceed with the lecture proper, there are some important terms and concepts that must be reviewed. So let's start with the term haploid. In here, it refers to the condition where a cell has only one copy of every chromosome. I will illustrate that more further here. So if you can still remember, the term ploidy refers to the number of each type of chromosomes. So this green circle here represents a cell and the nucleus where you can find the chromosomes. Now since the cell is a haploid cell, there must only have one copy for every chromosome in it. Okay, so I'll put here a long chromosome and a short chromosome. Okay, so each of this we have two different types of chromosomes, but each has only one copy. Therefore, this is a haploid cell. We usually refer to this as N, or we have N number of chromosomes, or one copy of each chromosome. On the other hand, the diploid condition is where a cell has two copies of its chromosome. So in the same manner as we did with the haploid cell, in here we should place two copies of each chromosome type. So in here I put two long chromosomes and another two short chromosomes. Okay, so we use this 2N to refer to the diploid condition. Let's review mitosis and meiosis. So from your previous discussions with your teacher, mitosis is a type of nuclear division that results in exact copies of the DNA being distributed to two daughter nuclei. It is usually followed by cytokinesis or cytoplasmic division, and it maintains the ploidy, meaning the number of chromosomes in the mother cell is conserved in the daughter cells. So here, for example, we have a diploid cell, and prior to mitosis, this diploid cell must first undergo DNA replication. That's why we have the 4N configuration right there. And eventually, this will split into two daughter cells, restoring the original diploid configuration. On the other hand, for meiosis, it is a type of nuclear division that results in a reduction of the chromosome number, and it involves two rounds of cell division. So if you reduce the chromosome number, you usually cut the ploidy in half. Okay, just like mitosis, you have DNA replication prior to meiosis, and after the first round of cell division, you get two diploid cells. Okay. And on the second round, there is no more DNA replication that takes place. That's why you eventually reduce that number into the haploid condition. In sexually reproducing organisms, we have a fusion of gametes at fertilization to produce a zygote. So we have an egg here, and of course the sperm. Both are haploid cells. Now we refer to these haploid sex cells as gametes, and they are used in reproduction. Now the fusion of these gametes is what we refer to as the process called fertilization. Now once you have the fusion of gametes, the nuclei will also fuse, resulting to a diploid configuration or a diploid cell. So this is referred to as the zygote, the first cell that is a result of the fertilization event and is therefore diploid. So there are two more vocabulary that must be defined. So we have the sporophyte, which refers to the plant generation with diploid chromosome number, and the gametophyte, which refers to the plant generation with haploid chromosome number. So shown here is the typical life cycle of a flowering plant, and this is described as an alternation of generations. So you have here an alternating haploid and diploid phases. So if we start with a mature plant right here, it undergoes gametogenesis, of course, to produce the gametes, the sperm and the egg inside this ovule. So it is during this phase wherein the plant is in its haploid state. Okay, you only resume the diploid state once you do fertilization, wherein you form the zygote. So from the zygote to the embryo to the mature plant after further growth and maturation, this is referred to as the sporophyte phase. Whereas this blue here, blue arrows will represent the entities representing the gametophyte phase. Okay, so let's have this short activity. So get ready with your papers and ball pens, the one that I gave you. Okay, so please follow instructions. We will do this together. So we will have alternation of generations. First, draw a line. Okay, this will separate our haploid and diploid phases in the life cycle. So in here, you write haploid, and of course N. Okay, on the other side, you write diploid and 2N. Okay, so we have the representation of these two phases. Okay, on the left side, you write gametogenesis. Okay. While here on the right side, you write there fertilization. So we have to set this out first before we continue with the concept mapping. Okay, so we can start. So we first have our gametophyte. Okay, so gametogenesis signals the haploid generation to start. So our gametophyte refers to the pollen grains and the ovule. Okay, the pollen grains are our male gametophyte, whereas the ovule is the female gametophyte. Okay, we know that the pollen grain will give us the sperm, while the ovule will give us the egg, of course. So these two gametes will eventually fuse via fertilization, and once you have fertilization, you start or you restore the diploid configuration. So the fertilization will signal the start of the diploid phase or diploid generations. So again, from previous discussions, we form the zygote via fertilization. Okay, and we write in here, the zygote will eventually divide via mitosis to form the embryo. And eventually the embryo will further develop and mature to form the mature plant. Okay. Once you have a mature plant, it can again produce gametes, it can perform gametogenesis, and the cycle is repeated. Okay. Now the embryo is that tiny little plant that you can find inside seeds, while the zygote again is the first cell in the sporophyte generations. Okay, so we will also draw here the mature plant. Okay, remember that all of this entities all belong to the sporophyte generation. Again, the sporophyte generation is the diploid generation. Compared to the gametophyte generation, okay, your keyword there is gamete. You should always expect that the gametophyte generation is in the haploid state. Okay, gametophyte, gamete, haploid. So don't forget the keyword. So you continue with your work. You finish everything before we proceed with the next slide. Okay, so let's start with the next activity. Okay, in here we will review the reproductive parts of the flower. Okay, so I'll first draw here the female reproductive part. So draw it like this. Okay, so this part is called the stigma. Okay, this is the style. Immediately below the stigma and the ovary, the enlarged portion. Inside the ovary are your ovules. Okay. Okay, the stigma is the receptive site for your pollen. This is where pollen lands to fertilize the egg later. The ovary will eventually become the fruit if your flower develops into a fruit, and the ovules will become the seeds. Okay, that is where you can find the embryo, as we have discussed earlier. Okay, so all of these structures are collectively known as the carpel, or the female component of the flower. Now we go to the male component. So we only have two parts or two components. We have the anther, where you can find the pollen grains, and the filaments, or the filament. The filament raises the anther so that it can be accessible to pollinators. Okay, this is the male component, or the stamen. So your clue there is the stamen. That's the male part. Okay, so does this look like a flower? So we add some structures or parts that will make this look like a flower. Okay. So we add some sepals and the petals, of course. But again, take note that these structures are not reproductive structures. They only facilitate reproduction, especially the petals, by attracting pollinators. Okay, so you draw your flower and label it properly. Label the reproductive parts. Okay, again, not reproductive. Okay, so continue with your work. Make sure you have completed all of this, labels and structures. Okay. Okay, so let's start with the third activity. We will review pollen formation that was discussed earlier. Okay, so let's have the anther. Okay, at the tip of the filament is the anther. This is where you can find the pollen. Okay. Now, if you look inside the anther, you'll find there the microspore mother cell. It's a diploid cell. Okay, so we have here the microspore mother cell. Okay, you do the same too. Okay, so this will divide via mitosis, so if it, or via meiosis, I mean, so to produce 4 haploid cells. Okay, 3, 4. Again, the microspore mother cell divides by meiosis to form 4 haploid microspores. Okay, 4 haploid microspores. Take note, there is a difference between the mother cell and the microspores. The microspore mother cell is the mother of the microspores. So each of these will undergo mitotic divisions, okay, to form the pollen grain, which is comprised of 2 cells. Okay. So we have there the tube cell, and inside the tube cell is the generative cell, meaning the tube cell is enclosing the generative cell. Alright. So this is your pollen grain. Okay, so let's have this final activity. We will review ovule formation. Okay, so let's have here the female reproductive component of the flower. Okay, and you're aware that we have ovules inside this ovary. Okay, so the ovule eventually or actually contains the diploid cell that will form the female gametophyte. So inside this is the megaspore mother cell. Okay, just like the microspore mother cell, this megaspore mother cell will divide via meiosis to form 4 haploid megaspores. Okay. But in this case, we have unequal cell divisions. 3 of the haploid megaspore cells are smaller, 1 is bigger. Okay, so we label this as 4 haploid megaspores. Okay, so these 3 here will eventually degenerate. So only 1 will be left to continue the process of gametogenesis. Okay, so what will happen to this? Of course, it will divide via mitosis, just like the microspore. Okay, you have mitotic divisions, and you will be doing this 3 times. Okay, so you expect how many cells? So on your first division, you will have 2. On the second mitotic division, you will have, of course, 4. cells, and on the third mitotic division you have 4 times 2 equals 8 cells. Alright, so you'll form 8 haploid nuclei. Okay. So where can you find all of this? They are found inside the ovule. So we have here a structure, a rough structure of the ovule containing the 8 nuclei. Okay, 1, 2, 3, 4, 5, 6, 7, 8. Okay, that is the ovule. Okay, 2 of the nuclei will fuse to form a diploid cell. So 2 of the 8 will fuse to form the diploid cell, whereas 1 of this 8 haploid nuclei will develop into the egg cell. So if 2 of this fuses, you eventually get 7 cells inside the ovule. Right. So 1, which is 2n because of the fusion, will be formed right there at the central part of the ovule. We have 3 of the remaining haploid cells, and 1 of these cells will become the egg. Okay, that's the egg that will eventually be fertilized. And we also have the diploid cell, which will also be fertilized eventually. Okay, so all in all we have 7 cells inside the ovule. Okay, so continue and complete all your illustrations there. Okay, I'll give you time to do that. Copy.
Sexual reproduction in angiosperms involves the transfer of pollen from an anther to a stigma, a process called pollination. In this example, pollen from the anther of one flower is transferred to the stigma of another flower. After the pollen grain lands on a receptive stigma, it germinates, producing a pollen tube. The pollen tube grows down through the tissue of the style. At some point during its journey, the generative cell of the pollen grain divides by mitosis to form 2 sperm nuclei, or male gametes. The pollen tube continues to grow until it reaches the ovary. In most plant groups, fertilization is straightforward. 1 sperm nucleus fuses with the egg to form a diploid zygote. In angiosperms, however, an unusual event called double fertilization takes place. The other sperm nucleus fuses with the 2 nuclei of the central cell to form a large triploid cell. The cell resulting from this second fertilization begins a series of mitotic divisions that forms a tissue called the endosperm. The function of the endosperm is to store nutrients.